This conference will now be recorded.
I am Sharmila Dorbala from Brigham and Women's Hospital in Boston, and I'll be your moderator today. I'm delighted to introduce to you the co-moderators of this session, the co-organizers of this session, Dr. Mehran Sadeghi from Yale University and Dr. Woonan Zhu from the NIH. Before we get started, just a few housekeeping tips.
Sharmila,
Please mute your microphones, all attendees. We will have a question and answer session at the end of each presentation by chat feature.
We do not see your screen, Dr. Dorbala.
You're not seeing the screen. How about now?
Yes, we can see it now.
All right, perfect. Thank you for that. So here are my co-course organizers, Dr. Mehran Sadeghi from Yale University and Dr. Woonan Zhu from NIH. Before we get started, a few housekeeping tips. Please mute your microphones. We will have a question and answer session at the end of each presentation using the chat feature. To post questions, please post questions using the chat feature. This presentation will be recorded and available on the ASNC website. Today, we are pleased to welcome attendees from institutions across the globe. The spring schedule includes a variety of topics focusing on atherosclerosis, new technologies, vascular calcification, transthyretin amyloidosis, cardiovascular inflammation, microvascular dysfunction, and more. The topic for this week is atherosclerosis, and part one of that will be presented today. Our agenda today includes a talk followed by a discussion of questions submitted via the chat feature. And after that, we'll have a research presentation on atherosclerosis followed by a discussion of questions submitted by chat feature. Today, we have joining us two outstanding speakers and world authorities on atherosclerosis. Our first speaker is Dr. Peter Libby. He's a cardiovascular specialist at Brigham and Women's Hospital and Mallinckrodt Professor of Medicine at Harvard Medical School. Dr. Libby needs no introduction to the field of atherosclerosis. He has served as the Distinguished Chair of Cardiovascular Medicine at Brigham and Women's Hospital from 1998 to 2014. He has pioneered several discoveries on the role of inflammation in vascular disease and atherosclerosis. He has translated basic lab studies into pilot studies in large-scale clinical cardiovascular outcome trials, the most recent of which is the CANTOS study. Dr. Libby has received numerous awards, including the Gold Medal of the European Society of Cardiology and other awards, and he has published extensively in several high-impact journals. I would like to mention two of his achievements that are not listed here. For those of you who do not know him or who know him, he is an avid runner who ran many marathons in several countries. Next, many of you may have heard about the unique multimodality integrated cardiovascular imaging program at Brigham and Women's Hospital. Dr. Libby was one of the founders of this philosophy of integrating multimodality imaging between cardiology and radiology, along with Dr. Seltzer and Dr. Marcello Di Carli. Now, coming to our second speaker, Dr. James Thackeray. He's a research group leader at Hannover Medical School. He trained at Ottawa Heart Institution and then went to Hannover as a postdoc in nuclear medicine and cardiology. His research interests include assessing local changes and interconnections in inflammation, fibrosis, and sympathetic neuronal activity after ischemic injury, developing a paradigm of imaging-guided therapy to optimize target and timing of intervention to improve functional outcomes, evaluating systems, biology, and interorgan communications via total body PET imaging to explore the systemic progression of disease. Dr. Thackeray is an accomplished researcher who has received many awards. He has received the Hal O'Brien Rising Star Award, the Jared Gwan Hevesy Award, the Jack Young Author Achievement Award, and the Young Investigator Award at SNMMI and ICNIC. He now has an established research team of his own with international members in it and includes 3 PhD students, 1 clinical fellow, and 1 postdoc, all of whom have won their own awards. Our discussant today is Dr. Mehran Sadeghi. He is professor of medicine at Yale University School of Medicine and staff physician at the VA Hospital in Connecticut. Dr. Sadeghi's research interests include vascular and valvular biology and molecular imaging, with an emphasis on vascular remodeling, angiogenesis, and inflammation. Dr. Sadeghi received many research awards, including the Nycomed Amersham Clinical Research Award from ASNC, Harry B. Graff Career Development Award for Heart Disease Prevention, and the Herman Blumgart Award from the Cardiovascular Council of the SNMMI. So with that, I would like to now invite Dr. Peter Libby to present his talk. Thank you. Joe, can you please make Dr. Libby presenter?
Sure. I believe Dr. Libby is still having connectivity issues. Should we start with James Thackeray?
Sure, that's fine.
Okay, one second. I will make you presenter. I can see your screen.
Okay, thank you very much. So I suppose I have the dubious honor of starting this then, which is positive in that I do not have to follow Dr. Libby's presentation then. What I wanted to do today is give a little bit of a flavor of the work that we do in Hannover, Germany, looking at my research group specifically in translational cardiovascular and molecular imaging. So just to first introduce us, because I have frequently been mistaken to be working in New Hampshire or in Pennsylvania or various other places in the United States, Hanover is located in the northern part of Germany. We are the 13th largest city in Germany. This is Hanover from Google Maps, and our hospital is located here just to the east of the Island Rita, the city forest in the middle of the city. This is our hospital campus, and our nuclear medicine facility is actually located basically in the center of the complex here as well. It's the only dedicated medical university in Germany. We have 8,000 employees, many of whom are back to work now, and 3,500 students, both within medical students and in basic biology and in physical sciences. We are a super maximal care facility with 1,400 inpatient beds, many of which are now being released out of infection wards, as well as specialization in research in immunology, in infection, transplantation, regeneration, and biomedical technology. We are consistently ranked in the top 5 in Germany in terms of biomedical research. Our department has actually fairly extensive history in molecular imaging. Hannover Medical School is the site of the first human MRI scan that was performed in Germany was performed here, and our small animal facility was actually built in the location where that MRI facility was. We work off of a paradigm of working from basic research, from molecular probe development through preclinical research and into clinical research and patient care, with an eye towards our patient care. We're working from developing new radiopharmaceuticals, evaluating them in mice and in pigs, and then moving on to large or first-in-human studies as well. We do this with a cyclotron that is now operational for the last year, as well as small animal PET and small animal SPECT that have been running since I arrived in Germany in 2012. So as I mentioned, what I want to do today is give a little bit of a flavor of the work that we do in my lab. And I'll be speaking for a number of different people who now are working on these projects right now. When we talk about nuclear cardiology, we typically are talking about looking at perfusion and viability, evaluating cardiovascular function, or evaluating SCAR. And the challenges here from the present perspective is that these are generally observational measures. They provide limited opportunity for a novel intervention against what is effectively a fixed defect. So imaging in terms of therapy is spent trying to evaluate how these fairly fixed defects respond to the damage or respond to the therapy. What we are looking at in our laboratory is moving beyond this conventional paradigm, looking towards more molecular imaging approaches. And the focus is then on image-based risk stratification, identifying which patients will benefit from therapy, using this to guide therapeutic intervention through image-guided therapy, and building on a systems biology response to look at how different organ injuries affect more systemic biology. And we do this focusing on a number of different molecular imaging targets, some including inflammation, which is fairly ubiquitous, fibrosis, and innervation. And if we look across the gamut of the healing perspective on the heart, what we work on predominantly in the moment is myocardial infarction and ischemic injury, not so much in the atherosclerosis end, but the targets are in some cases rather similar. We can look at moving from inflammation in the initial injury through fibrosis and the healing process towards remodeling. And what I'll focus on today is three specific compounds. That is the chemokine receptor CXCR4, which is expressed by a wide variety of different leukocytes, the translocator protein TSPO, which can be imaged with a wide range of different imaging agents, which targets the translocator protein in mitochondria, which are expressed in peripheral macrophages as well as microglia, and the fibroblast activation protein, which is part of the involved process in mediating the transdifferentiation from fibroblast to myofibroblast and the formation of scar. So when we first look at inflammation, following a myocardial infarction, there's an organized infiltration of activated leukocytes to the damaged region, characterized initially by more pro-inflammatory cells and later by more anti-inflammatory or reparative cells. And while we do a very good job of reperfusing the infarct and treating the long-term remodeling processes, there's an underexploited therapeutic window, which is characterized by this pro-inflammatory phase that can be targeted with novel therapeutics. And this is where molecular imaging may be able to identify differences in the inflammatory response between individual patients. We've started looking at this predominantly with a CXCR4 ligand, so a chemokine receptor for CXCR4 ligand, gallium-labeled pentixafor. What we can see in these images, this is mice following a permanent occlusion of the coronary artery. We see a upregulation of the CXCR4 signal coming from the infarct territory. This is in the grayscale defined with FDG, so the absence of FDG uptake is the scar area. We see a high uptake at day 1, which dissipates a little by day 3 and is basically back to baseline around day 7, compared to the sham where we don't get this elevation at all. You can see that in the time course as well. Upregulation at day 1 and day 3, reduced by day 7. And this lines up very well with flow cytometry data, where CD45 positive leukocytes follow the same kind of pattern with an early upregulation that is then mitigated by day 7. We've used this information to try to guide or identify how the inflammation actually influences the long-term outcomes in these animals. So if we look specifically at the upregulation of CXCR4, the CXCR4 signal at 1 hour and 3 days, we can actually predict the outcome in these animals later on. So the pentixafor signal as early as 3 days after MI is the higher the pentixafor signal, the more CXCR4, the worse these animals do 6 weeks later. We end up with a lower ejection fraction as defined by MRI and a decline based on higher inflammation, worse functional outcome 6 weeks later. We use this information to set up a paradigm of image-guided therapy. So effectively, we looked at the images, we identified a time where we have an upregulation, this is at 3 days, versus a time where we don't have an upregulation at 7 days. And we treated these animals with a FDA-approved treatment against CXCR4, an inhibitor, AMD 3100. So with an on-peak treatment or an off-peak treatment. This is work by PhD student Annika Hess in my lab. What we observe is there's an immediate, with the AMD 3100 treatment at the time of high elevation, we get a reduction of the acute ventricular rupture occurrence and acute death in these animals over the first 7 days, which is akin to the clinical expansion of the infarct. With a permanent occlusion, you end up with left ventricular rupture in mice. And what's more interesting is that by 6 weeks, the animals that do survive have a better contractile function. So the ejection fraction is actually spared in the animals that are treated at the appropriate time. If you treat them when the CXCR4 signal is not elevated, you don't get that same response and the ejection fraction does not go up. can actually be applied in transverse aortic constriction or in pressure overload-induced heart failure as well, where you can see when we co-localize to the FDG signal, there is an upregulation of CXCR4 as well throughout the left ventricle. And this signal also predicts the functional outcomes. So to higher the Pentixafor signal at 7 days, 3 weeks later, we get a larger systolic volume. So we can use this information to identify how the animal or how the patient is going to respond in the long run. And interestingly enough, the Pentixafor signal also parallels T1 relaxation time on MRI as a surrogate marker of fibrosis, which leads us into our next point, which is looking at fibroblast activation protein. This is work by Laura Lager in our radiochemistry department, looking at fibroblast activation protein as a marker of the activation and transdifferentiation of fibroblasts to myofibroblasts that then form the scar. What we observe is that within the first 7 days, there is a clear upregulation of the fibroblast activation protein localized to the infarct territory. It co-localizes very nicely to where the fibrosis is defined by mass and trichrome, so the area that's blue, is pseudo-co-localized to the CD68 positive macrophages, but the CD68 positive macrophages don't co-localize with the fibroblast activation protein. That protein is expressed on these transdifferentiating fibroblasts. This is of particular interest because therapies that are targeted to fibrosis are emerging as possible treatments for myocardial infarction. Then the other area that we're interested in is looking at how the injury to a specific organ influences the rest of the body. The seminal work that we did on this published in 2018 looked at inducing a myocardial infarction in a mouse, so a coronary ligation, and then we imaged with this TSPO marker that's traditionally used for neuroinflammation, so it's taken up by activated microglia. And what we observed is that while we have an upregulation in the infarct territory of invading peripheral macrophages, at the same time, in the acute phase, we see an upregulation of TSPO positive microglia in the brain and throughout the brain, which recurs when these animals reach chronic heart failure. So the signal in the heart goes from the infarct territory, and then it migrates out to the remote territory as we start getting mitochondrial dysfunction and failing myocardium. But the neuroinflammation responds directly to the cardiac signal. We were able to repeat this with a different tracer. This is C11-methionine, where we see the upregulation of C11-methionine in macrophages enriched in the infarct territory here in the color scale, which parallels an increase in the global brain signal as well. So again, the brain signal responds to the infarct. So a local injury to the heart influences brain inflammation. And this actually tied more to the astrocyte activation into microglia, suggesting that we can actually identify different phases of the neuroinflammatory response following myocardial infarction. We have just started work in ischemic stroke to look at the reverse process of this phenomenon. So basically, instead of the heart-brain axis, looking at the brain-heart axis. So Nayla Hermans in my lab has developed a model of middle cerebral artery occlusion, where filament is advanced through the cerebral artery, blocks the middle cerebral artery through the carotid. And then we looked at the initial inflammatory signal coming from the stroke territory as defined by the MRI. We observed that in the ipsilateral hemisphere, there is a clear upregulation at 7 days, and at 21 days, it remains elevated compared to the opposite side. But what is interesting is you actually get cardiac inflammation, or you get an increase in the TSPO signal coming from the heart in response to the stroke injury, which reflects a systemic inflammatory response to the ischemic damage. And this also correlates with a 3-week decline in left ventricular ejection fraction. And this work of Nayla's will be in the Young Investigator Award session at the SNMMI in the virtual meeting later this year. The last point I wanted to talk about is that the healing of the myocardium actually influences the brain as well. So in a study by Tobias Boyhert, who just finished his PhD in my group, we depleted macrophages by giving them claudinate-loaded liposomes, which kills the peripheral macrophages. So we clearly see that the infarct territory shows much lower signal than we get in an untreated MI animal at 7 days. But these animals actually see an increase in the brain signal in terms of microglial activation in response to the myocardial ischemic injury. There is a lower acute survival, so the rupture rate goes up. The ejection fraction is a little bit worse, but this rebound or increase in the brain neuroinflammatory signal actually suggests that the lack of macrophages to clean up the neutrophils inside the early wave of the infarct injury results in higher cytokine, pro-inflammatory cytokine mobilization to the brain and a local inflammatory response that is enhanced. So what we're focused on is looking at beyond our conventional imaging markers, looking beyond these markers to look at molecular imaging targets of pathophysiology, which are ideally then merged or coupled to therapeutic targets. So with CXCR4 or with TSPO, where we can treat what we're actually imaging and be able to titrate doses appropriately. We can move beyond diagnostic imaging in terms of looking at the early inflammatory signal to predict the functional outcome, allowing us to move towards more of a precision medicine paradigm to identify the right patient for a particular therapeutic intervention, moving us beyond focused pathophysiological consequences towards looking at how different organs interact and different diseases interact, from inflammation to fibrosis, and then beyond organ-specific imaging, moving towards this concept of total body PET, which we have formed a strategic partnership with Davis to start looking at doing this in patients as well, and inter-organ communication in disease and treatment. So that's a basic overview of the work that we're doing in Hanover, and I'm happy to take any questions.
Thank you, James. It was an outstanding presentation and raised a lot of important issues. You overviewed potential application of molecular imaging in cardiology. We do mostly perfusion imaging, and now this is really the future of how we can use nuclear cardiology and molecular imaging in our practices. So how far are we from translating these findings? The tracers that you used, you mentioned at least 3. I know some of them have been already used in humans. Can you just give some information about that, and what are the barriers to the translation?
So we do have the benefit in Germany of having a little bit of a more streamlined approach to doing first-in-man studies. So all 3 of these compounds that I've talked about today, the FAPI46 for fibroblast activation protein, CXCR4 Pentixafor, and our GE180 TSPO, have all been done in-house in clinical studies. There is still there will be a challenge to trying to get larger scale studies in the long term and trying to roll this out beyond Germany, which has its own unique architecture to allow us to do these. But by using the experience in Germany in smaller scale studies, we can actually get the preliminary data that allows us to do larger scale studies and show that there is an actual added value. So I think that's part of the challenge.
Great. So the audience can send questions by chat, and we'll monitor them and ask Dr. Thackeray their questions. So the questions from the audience is really welcome. A question from audience before I ask another question. Can you please, how essentially this FAPI tracer is working in the myocardial imaging? Can you comment more about fibrosis imaging and how this thing works?
So we've just started with FAPI46. There was a paper that came out earlier this year with FAPI04, which specifically shows the upregulation of fibroblast activation protein in myofibroblasts after the ischemic injury. So the time course or the evolution or the physiology of fibroblast activation protein is it's expressed at a very low level by fibroblasts to begin with. When they are activated in an injury situation, you see an increase in the fibroblast activation protein expression. And that's what you're targeting with the FAP inhibitor tracers, of which I think, well, if there's 46 of them, there's at least 46. There's a wide range now. So we see the upregulation in on our autoradiography sections. We can see it colocalizes quite nicely to where the scar is forming. We are starting to look at which cells are actually expressing the FAP directly, but we haven't gotten to that point yet.
And these tracers, they've been also applied to oncology now. So there is some body of literature that is being formed in that field.
Yes, there's a wide range of stuff that we are learning from oncology. I think the whole idea of trying to build these image-guided therapeutic ideas really builds on the experience from oncology of coupling the imaging agent to the therapy so that you could actually have a pseudo-theragnostic approach, not necessarily with radiation therapy, but being able to look at the expression profile of a specific marker. CXCR4 is a great example because the patients that we have with CXCR4 post myocardial infarction, there's basically a broad range of CXCR4 expression. There are patients with virtually no expression and patients with a very strong signal. We think that we would then be able to identify patients who would respond to that type of therapy a little bit more effectively. But that is work that is still in progress. We have a series of about 90 patients that's just finishing up the follow-up, which will be interesting to see how that all works out.
So while I'm waiting for more questions from the audience, so comparing cardiology with oncology, in oncology, you remove the tumors and you can do a lot of validation studies. In cardiology, this is going to be much more difficult. So how do you foresee to approach this, essentially these important validation studies?
Yeah, that's always going to be the challenge from the cardiology end, is that we do really well with the drugs that we have. And trying to identify whether the molecular therapies are having a better effect is also going to need to define the right patient populations to be receiving them in the first place. I think it's a formidable challenge. It's a little bit outside of my laboratory as to how we're going to be managing this. But I think the important thing is going to be working from these streamlined kind of ideas, from developing the compounds and testing them preclinically to move into the clinical application, to keep an eye on how this can actually benefit patient care. So I don't think that actually answered your question particularly well, but
no, but that's good. I mean, that raises the challenge that we have in cardiology to translate these tracers. And while we are continuing, maybe they can give, Dr. Libby is getting prepared. I think we can present the slides on the screen to Dr. Libby. So I have another question in this regard, James. So this inflammation, role of inflammation in the atherosclerosis, in myocardial infarction is very complicated, and there are different subsets of macrophages. So the tracers that you mentioned, the two, TSPO and CXCR4 pentoxy-4, what do you think they're targeting, and is there a challenge to know what's the right timing?
Yeah, without question. So with pentoxy-4, for example, we've actually, with all of these compounds, we've actually tried to do preclinical screening and in vitro screening to see how the distribution in different cell types actually, how it differs between pro-inflammatory, anti-inflammatory, various types of leukocytes. The expression profiles tend to be pretty broad, and what happens in a petri dish is not what happens in real life. What I think the benefit of pentoxy-4 that is a little bit more broad is expressed by this wide range of different cells. The benefit of pentoxy-4 is that it's really giving you an idea, from what we can tell, of total number of leukocytes that are there. And whether or not you have this, we do have this movement from pro-inflammatory to more reparative, if we view it in that way. The total number of cells still matters. And the pro-inflammatory wave is dominated by more number of cells than the reparative wave is. So if you are able to identify that early phase, and the early phase is where you actually have, I think, the clinically relevant therapeutic window to actually have a benefit in post-myocardial infarction.
So James, there's two questions, James and Mehran, both two questions on the chat. One was about gallium-68 fibroblast activation protein. What is its role in myocardial imaging? The second question is, how could we get a reliable target out of all the potential targets so that we can figure out which one is an important fundamental therapeutic target?
Yeah, so the first one, fibroblast activation protein, is involved in the transdifferentiation of fibroblasts to myofibroblasts in the formation of scar in the development of interstitial fibrosis. So we're starting to look at it in the TAC model as well. It's integrated into that whole remodeling process. For getting a reliable target, I think that's been the challenge with a lot of nuclear cardiology novel tracers, is we need to be able to identify the strong candidates early. And by building more interconnections between different research arms, we will have a better idea of how to develop these more efficaciously. Instead of having 30 different compounds that target different chemokines, we need to be able to agree which chemokines are the right ones to actually be targeting. And that's a challenge. It requires us to actually have more conversations like the ones we're having. And I think that's building these partnerships and building the networks that allow us to test these outside of individual centers is going to be very important for building towards that goal.
Thank you, James.
Thank you, James. That was a great presentation, and that I think that Dr. Libby is now, I saw his slides, and I think we have time to move on to Dr. Libby.
One second, I'm going to make him presenter. Dr. Libby, you should be presenter now. Let me know. Oh, actually, one second.
Yeah. So, you know, every time that I try to share my screen, it crashes the app. So okay. I will try again on my laptop and see how that works.
Okay. Are you on your laptop now, or should I wait for you?
No, I'm on my laptop now. You know, this is why I wanted to use Zoom, but Zoom is not allowed in Germany because Zoom actually works. Okay. So how do I share my screen?
Okay, you should be presenter now.
Yeah, we see your screen, Peter.
Okay, good. Okay, great. Sorry for the technical glitches. As I say, I use Zoom for about 4 or 5 hours a day, and it works flawlessly, but we had some constraints with it. So thanks very much. Actually, James' talk was perfect introduction. And I'm going to talk about atherosclerosis pathophysiology and try to finish with some implications for molecular imaging. Can you hear me okay?
Yes.
Good, okay. These are my competing interests. So, you know, atherosclerosis is a disease probably with the longest incubation period of any human disease, beginning actually in youth and progressing until the ultimate clinical manifestations. The normal artery wall, to review, consists of 3 layers. There's the intima with its lining of endothelial cells, and in the human intima, as opposed to the mouse, we do have resident intimal smooth muscle cells, the tunica media and the adventitia. Atherosclerosis begins when we recruit leukocytes, notably the leukocyte that's most involved in chronic inflammation, the mononuclear-phagoside. We've already talked about chemoattractants that cause a directed migration of those cells into the intima. The monocyte will mature into macrophages, express scavenger receptors, and mature into a foam cell, the hallmark of atherosclerosis. This slide is 18 years old and has been widely copied and actually has held up fairly well through the years, where we were fortunate to be able to work on some of the initial characterization of the molecular mediators of those various steps. The chemoattractants and their receptors, scavenger receptors allow the capture of the lipoproteins that lead to the formation of the foam cells and, of course, many of the mediators that cause the complication of the plaque and ultimately its clinical manifestations. Please don't pay attention to this slide. It is about the fourth generation of simple slide of the immunology of atherosclerosis. I want to make one point with this slide. I refer you to this review if you're interested in more detail. And that is that we have crosstalk between innate immunity and adaptive immunity that really is a yin and yang, some positive influences, some inhibitory influences on atherosclerosis that probably account for this very long incubation period of this disease. For those who'd like to come up to speed about some of the current thinking about atherosclerosis, probably at the level of a beginning graduate student for those who work in other domains, I refer you to this recently published review in Nature Primers. And this is one of the figures from that review, which tries to convey this idea of the yin and yang, or the tug of war between life and death, between pro-proliferative and anti-proliferative, pro-inflammatory, anti-inflammatory, and pro-resolving mediators. So there's this contest that's going on constantly, and this does give us some targets for imaging. There's been a great deal of interest in the mechanisms of the thrombotic complications of atherosclerosis that bring the patients to our attention most dramatically in the clinic. And this one slide represents probably a quarter century of work from my laboratory, looking at the factors that regulate the integrity of the plaque's fibrous cap, which largely is governed by interstitial collagen. And we've gone to great lengths to look at the way in which the synthesis of collagen can be inhibited by pro-inflammatory cytokines that interferes with the maintenance of that all-important fibrous cap. That's all that stands between many of our patients and acute thrombotic complications of atherosclerosis. We also spent a great deal of time working out the regulation of proteinases, particularly the interstitial collagenases that are members of the matrix metalloproteinase MMP family that can degrade this usually very stable molecule. And so we've gone a great deal of the way towards understanding in precise molecular terms the features that govern the propensity of an atherosclerotic plaque to rupture. However, I would like to be a bit provocative in several ways today and pose the question, is the quest to image the so-called vulnerable plaque a fool's errand? And I wrote up with Gerard Posterkamp some of my thoughts about this a few years ago. Why do we believe that this idea of the vulnerable plaque is challenged? Because our risk factor profile has changed. The demographics of our ACS patients have changed, and human plaques in an era of control of lipids, blood pressure, and reduced smoking has changed. Back in 1995, I hypothesized that if we lowered lipids stringently, that we would reinforce the fibrous cap, reduce the lipid core, and indeed, that's what we're seeing. In the human imaging studies, this is MRI studies and intravascular ultrasound studies with intensive statin treatment, which is now becoming the norm in patients at high risk. We reduce the lipid core, we increase the fraction of the volume of the plaque that's made up of fibrous tissue, and we actually increase calcification. So think about that, you people who do calcium scoring and patients on statins, that calcification actually increases with statin use as events decrease. There's an inverse relationship. With Gerard, who has a very large tissue bank, we were able to look over time at the structure of human atherosclerotic plaques as these secular changes in risk factors and in medications have been occurring. And what we can see is marching through in time that there's less inflammation, much less, more than halving of the lipid core content. So the classical so-called vulnerable plaque, the thin cap fibroatheroma, is actually passé. And we always tend to face today's battle prepared to fight the last war. And so I'd like to challenge people by thinking if we really should be chasing after the vulnerable plaque in the current era. And one of the big challenges to this concept clinically is an imaging study. This is Greg Stone's PROSPECT study, where they used virtual histology to characterize plaques and followed people up for greater than 3 years, and found that 95% of the plaques that are so-called vulnerable plaques cause no clinical problem in over a 3-year follow-up period. So vulnerable plaque is a misnomer. Over the last 5 years, 5 or 6 years, my laboratory has turned because we believe that we've solved this problem both clinically and from a scientific perspective towards plaque erosion. We think that that is an area of considerable scientific opportunity going forward. So if you want to learn about plaque erosion, which also has been really advanced by imaging, particularly optical coherence tomography in humans, you could read this article. Now, we know that inflammation is a driver of many aspects of atherosclerosis, and I don't want anyone to think that inflammation and traditional risk factors are in competition. Inflammatory pathways actually provide a series of avenues by which traditional risk factors and emerging risk factors can alter arterial wall function. So there's not a competition. It's a false dichotomy to say lipids versus inflammation. Actually, lipids probably work in part by inciting inflammation. And there are non-traditional risk factors. We have a great deal of interest. We're doing a large clinical trial now with targeting triglyceride-rich lipoproteins, a great deal of interest in ectopic adipose tissue, the microbiome, and most of my lab now is working on clonal hematopoiesis, which is a very recently described independent risk factor that is age-related. Now, how do we talked about the valley of death in molecular imaging. How do we translate all of this inflammation biology to the clinic? Well, we have to do clinical trials. And we focused, based on 3 decades or more of work from my laboratory, on interleukin-1, a primordial pro-inflammatory cytokine. I refer you to this article for the basis of the clinical trial that we undertook to test the inflammatory hypothesis in people. This is the design of the trial, which used a monoclonal antibody to neutralize interleukin-1 beta. We looked at hard endpoints, and we selected people who had survived the myocardial infarction, were on full menu of standard of care, but still had some residual inflammation as gauged by above median C-reactive protein. The intervention did not change atherogenic lipoproteins, but was handily anti-inflammatory. And we were able for the first time to show in humans that an anti-inflammatory intervention could reduce major adverse cardiovascular events. We paid a price in terms of host defenses. There was an increase in infections and a small increase in fatal infections. That was counterbalanced by a remarkable reduction in fatal malignancies, notably lung cancer, because our selection criteria enriched the population for people with lung cancer. Now, we did meet our primary endpoint with targeting IL-1 beta. What about other anti-inflammatories? Well, methotrexate has transformed the practice of rheumatology and dermatology. And so Dr. Ritker, my clinical partner, undertook a study, the cardiovascular inflammation reduction trial, to see whether treatment with methotrexate could also reduce cardiovascular events. The trial was halted prematurely for futility, but interestingly, as opposed to canakinumab, there was no anti-inflammatory effect with methotrexate. So we were able to have a positive study targeting IL-1 beta, but a null study with low-dose methotrexate. Very recently, a lot of interest has turned to using a natural product, colchicine, as an anti-inflammatory agent, and the Colcott trial was the first large-scale randomized double-blind placebo-controlled trial that showed efficacy of colchicine in myocardial infarction survivors. That study has spawned a great deal of interest, and there are many studies that are underway now, some of which will report, particularly the LODOCO-2 at the virtual European Society of Cardiology at the end of the summer. The spectrum of anti-atherosclerotic therapies is pretty broad, but the problem is that everything works in preclinical studies, everything works in the pilot clinical studies, but most of these therapeutics crash and burn when you do the large-scale, properly powered clinical trial. For those who are interested in the future of anti-cytokine therapy, I refer you to this fairly recent review that I did with Sadeghi and Hafid et Boufela. But to make it simple, beyond the targeting IL-1, we can mine that pathway because the methotrexate pathway looks less interesting. There's colchicine, there's inflammasome inhibitors, there's the anti-IL-6 pathway, etc. So I've spent my life studying vascular inflammation, and who knew that now in the spring of 2020 that we would be facing a really rip-roaring vascular inflammation in the cytokine storm, which is killing people with COVID-19 disease. So it's interesting how things go around and come around, because I think that these same anti-inflammatory interventions, which we've studied in the cardiovascular arena, are being studied now in the over 900, in some of the over 900 clinical trials that are currently trying to stem the damage of COVID-19. Now, I would like to finish with a couple words about molecular imaging. How can molecular imaging help us as we move forward? We already heard from James how our standard of care is so good, it's difficult to show an incremental benefit. And so we have to, as he said, in the direction of precision medicine, get smarter. We can use molecular imaging for dose selection, because getting the dose wrong is one of the major failure modes of clinical trials. To show targeted engagement early on in clinical development, to provide an initial indication by biomarkers of efficacy, and to select individuals likely to benefit from the intervention. That's the tack that we took in the CANTOS trial, which was successful by enrolling those who had a biomarker that was elevated, that was related to the mechanism or presumed mechanism of our intervention. So that's a way forward, and molecular imaging can help us there. With respect to the targets, everyone knows this, you guys know this better than I do. We have the ability to visualize many of the steps which I've spoken about. And in particular, because of the installed base of PET and the generation of fluorodeoxyglucose as part of oncology programs worldwide, there's been a great deal of interest in molecular imaging in the cardiovascular system with fluorodeoxyglucose. And it's amazing to me how little rigor has gone into the validation of FDG as a marker of inflammation. I think it's not so simple. I was trained in radioisotope methodology as a biochemist and learned that you cannot quantitate a biological process that you trace with a radio marker, with a radioisotopically labeled marker, without knowing the specific activity of the precursor pool. And because we have a rich microvasculature in the plaque, we, together with Viviane Taquete and Marcelo, have shown that actually we may be delivering the radioisotopically labeled glucose analog preferentially to areas of the plaque that are enriched in microvessels, and that would increase the specific activity. And so you can get an increase in radioactivity incorporation or uptake or accumulation without there being a change in the actual rate of the process. And as I'll show you in a moment, we tried very hard in my laboratory a decade ago to show that inflammation could turn on glucose transport in mononuclear phagocytes, and were unable to do so. But the first experiment with hypoxia was positive. So I think that a lot of what we're seeing with FDG uptake is looking at hypoxia rather than inflammation per se. And this commentary was accompanying an article from Sadeghi in Madrid that showed that smooth muscle cells could also contribute to glucose uptake. But the same study where we failed to show macrophage regulation of glucose uptake by inflammatory mediators, we showed that smooth muscle cells did respond. So here's that reference, which people just blithely ignore and equate FDG signals with inflammation with very little rigor in the biochemical thinking. We heard wonderfully from James about the cardio-brain axis or the brain-cardiac axis, and actually work that we did with Matthias Narendorf, Phil Swirsky, and Partha Dutta actually pioneered this area by showing that when there was a myocardial infarction, that it could potentiate inflammation remotely in the coronary atheroma. And we worked out the pathways that included the spleen. So there's not only a brain cardiac axis, but there's a splenocardiac axis. And it was actually molecular imaging that helped us validate that in humans. This is work that was a collaboration between Matthias and Ahmed Tawakol, where they were able to show that in acute coronary syndromes, not only was glucose uptake increased in regions of atheroma, but also in the bone marrow, where we have shown increased hematopoiesis, and in the spleen compared to controls. And here we can actually look now at markers of proliferation using the thymidine analog, showing that in ACS patients that there is an increase in bone marrow uptake of this indicator of cell proliferation. So you folks in the molecular imaging community can help us out in our cardiovascular therapeutics and diagnostics, dose selection, target engagement, early biomarker indications of efficacy, and very importantly, getting smarter in how we enroll patients in clinical trials to enrich for those who will respond. So I think I'll stop there and leave some time for discussion. Thank you.
Thank you, Dr. Libby. This is an outstanding lecture and covered a broad range of topics which are very relevant to molecular imaging and how we're trying to translate it. So I'm waiting for questions from the audience, but I already have one, and it's about, essentially you alluded to that in your last slide, investing in new tracers. There is a lot of, you need a lot of investment in new tracers, and it costs a lot of money. So do you think that pharma biotech would have interest in sort of seize the interest in using these tracers for dose selection, target engagement, etc., that you mentioned, and how do we sort of promote this more?
So, the radiochemistry is within the province of academic investigators. We just heard about that. GMP, the manufacturer of drug-grade radiopharmaceuticals, is a bit more of a challenge. And it's usually beyond the reach of a single academic lab, and that's why we have to have specialized centers that can do the radiochemistry and GMP. Then you have to do the clinical validation. You already brought up in your discussion some of the difficulties that we have compared to the oncology field in cardiology of doing validation. But there's been very little appetite on the part of the radiopharmaceutical industry or of pharma in investing in getting across the valley of death. So we've got great ideas. We can make mice glow in the dark, but actually getting a validated imaging agent that we could use for example, these 4 points on this slide has proven to be very difficult. One of the reasons that after trying to engage in molecular imaging and clinical translation, I decided it was too hard for me, so I left it to you guys, because it's just was very difficult to raise enthusiasm of the people who have the resources to support crossing that valley of death.
And in fact, we have a lecture on this on next week about this issue and how we can address these issues. There is a question from the audience about FLT that you mentioned. So can you comment on FLT, and do you think that FLT, the question is, do you think that FLT would do better than FDG for plaque imaging, and is there a role for that?
Well, I think the point of that experiment is that it we spent a dozen years or so looking at hematopoiesis. It's a major, clonal hematopoiesis is a major area of interest in my laboratory now and laboratories around the world. But a lot of that's based on in vitro and experimental work. And the wonderful thing about the thymidine analog is that it allowed one to actually test in people whether there was a signal in hematopoietic organs like the bone marrow. So I think it was a research tool that answered a very important research question about translatability of this large body of work on hematopoiesis in the mouse to the human. But I don't think that it's necessarily going to be a practical diagnostic or therapeutic guiding kind of technology. There's a great deal of interest. We had a wonderful grand round, Sharmila knows, from David Newby, who's in Edinburgh, who's interested in looking at what they believe is early calcification using fluoride, radioactive fluoride imaging agents. And that seems to be pretty promising. And because we don't have the background problems that we have with FDG, the signal is a lot cleaner. So I think we should keep our eyes on that if we want to look for novel, really immediately translatable plaque imaging modalities with PET anyway.
Yeah. Peter, thank you very much. Actually, David Newby's group will be presenting at this seminar series in maybe next week, right? I believe it's next week. So with that, I think we have just one more minute to go at this meeting. So I just want to provide two closing remarks. One is a big thank you and a big round of virtual applause for Dr. James Thackeray and Dr. Peter Libby on delivering really two outstanding and very insightful talks for this inaugural ASNC virtual molecular imaging series. So thank you again. And then the next slide I wanted to share with those of you who are in research labs and in training. These are the resources that ASNC has provided for researchers. We have a Young Investigator Award for basic and clinical science. The deadline for the submission is June 2020. So we encourage all research labs to consider submission of abstracts to the ASNC annual meeting. The second thing is ASNC has grants available, pilot grants for young researchers. There's one IANC research grant, which is awarded every year. And this year we have a specific grant on amyloidosis. Again, deadline for these two research grants is in June. Please apply, send your research to ASNC, get some research pilot grants that could help with your research. And of course, lastly, please send your publications to the Journal of Nuclear Cardiology, which is the journal of the American Society of Nuclear Cardiology, for publication. It will reach all the relevant readers on this. So with that, I want to thank everyone and just list the next session, which is May 14th, Thursday, 11 a.m. to 12 p.m. Eastern Time. The topic is Atherosclerosis Part 2. We have two research presentations on Thursday, one from laboratory of Dr. Pamela Woodward from Washington University in St. Louis, and another presentation from NIH, Dr. Nehal Mehta's group at NIH. So with that, I close this session. I want to thank my co-organizers, the entire ASNC staff, all of you for attending, and special thanks to the two presenters. Thank you very much. Have a good day.
Thank you.
Bye-bye. Thank you.
Bye, Peter. Thank you. Thank you, James.
Thank you.
Thank you.